seminar. We have one of our own speaking today, Xinyu Liu, I hope I pronounced it correctly. Yeah, Xinyu Liu from the Mechanical Engineering Department will be presenting on his work. Xinyu has already an amazing career. You came to U of T from McGill, I have seen. One cold place to another cold place. But I think we're slightly warmer. You've done a lot of Xinyu's done a lot of amazing work, received for all this work 10 Best Paper Awards, holds the chair for Microfluidics and biomaps. And I hope you'll explain to us what this means, because I'm new to this area and I'm super excited about this area. It has a lot of senior positions within IEEE Robotics and Automation Society, like the chair of the technical committee for micro and nano robotics and automation, and senior editor of Transaction on Automation Science and Engineering, and RAL, of course. And of course, I forgot, you're also a fellow of the Canadian Academy of Engineering. You have a lot of the very fancy achievements. Congrats. We're very excited about your talk. It's about robotic manipulation, a topic that we very much love, but our robots are pretty big and clumsy, and I think the things that you're manipulating are a little bit smaller than the things that we manipulate. It's robotic manipulation of small model organisms. We look very much forward to your talk.
Thank you, Igor. Thanks for the nice introduction. Thank you very much. Very nice to see many familiar faces and new faces through this event. I think I haven't done any talk since our formation of our RI, and it's very nice to report to everyone some of our recent research along this direction. And Igor mentioned the robotic manipulation. We have so many, fancy achievements by many other colleagues on large-scale manipulation. I would say when you get into small scale, there are some challenges, but overall, it's more structured in terms of the manipulation. So I'll introduce you some of the unique challenges we're trying to make, but overall, if you allow me to compare the large scale and the micro scale manipulation, I would say the micro scale manipulation has some of us is all unique challenges, but overall is a little bit easier. But I will show you some of the challenges, all right? Igor mentioned my research in terms of microfluidics and biomaps. In this work, I will show you some of our work involving robotics and microfluidics, especially for micro-scale biological optics. It's usually convenient or sometimes give you some advantages to use microfluidic tools. That's why we use those devices for sample manipulation or sample preparation. So I'll show you some of those examples. But on another line of microfluidics my lab is working on is microfluidic devices for diagnostics. That's like a separate line, mainly focused on portable devices for diagnostics purposes. But I will not talk about that direction for this in this talk. All right. So you see, I also add a term, characterization, in the title, because we manipulate those objects. We achieve some of the biological, we enable some of the biological processes, but at the same time, during those manipulations, we also do vision-based characterization. We do a 3D reconstruction of those micro morphologies of those objects, with the hope those information we gathered through those characterization could serve some biological meaning purposes. All right, I'll talk about that as well. So the object we're manipulating is small-scale organisms. We say model organisms. I'll give you some background on that. This is the outline of my talk. I'll give you a very brief background about this particular model organism, C. elegans, the nematode worm C. elegans. And then after that, I'll talk about a series of robotic systems and devices which we use to manipulate such a small animal. You can feel it lovely, but sometimes people feel disgusting because you see a snake type of locomotion. But anyway, the second topic I will do the second topic is related to a very traditional operation in the field we have been doing for a long time. Is we use a micro needle to do automated injection of those C. elegans. We also have other colleagues, like for instance, Professor Yu Sen, he's doing cell injection. And then here, I will show you something more locomotive. You will see it's a little bit harder to inject those guys. And then the third topic is related is during those injection and also raised by some other biological needs, we also do robotic or automated characterization of C. elegans. For instance, we measure their morphology, we do 3D reconstruction of their surface structures, and then we also characterize we'll perform high resolution molecule characterization of their neural systems and etc. But it all involves certain level of manipulation, automated manipulation. So the last one, maybe this is the oldest work I included here. Maybe some of you have seen some of those reports or some videos is in terms of manipulate a particular structure of those such a small animal. We also manipulate the whole animal. With the hope we can convert it into a robot. It's something crazy, but we never achieve perfect control, or we never achieve a complete conversion of such a small animal into a robot. But I will show you some the first work we did. All right. So, but overall, also those three topics are related to manipulation and characterization. So, C. elegans, you can see the left-hand side shows you a schematic. It's a very small worm. It's a worm-like animal. The length is about 1 millimeter. The diameter is about like 50 to 100 microns. It's a small worm. If you see it under the light, you probably can see it, but you have to rely on microscope. That's why it's a micrometer-sized object. And why we care about this guy? Because it's a very popular biological model organism. At least the full Nobel Prize is achieved in the past 25 years. They are all based on this small model organism. All right. So why do we use it? I'll give you some features of this guy. Is first of all, it has a very simple structure. This small worm only contains 1,032 cells, and if you think about the neural system, it only contains 302 neural cells. Their connection has been all mapped. That means you know exactly which neural cell is connecting to who. And then that's why it's an easy to use model to study neuroscience. This is a very popular research topic. And besides that, genetics. And this small worm doesn't look like human at all, right? But if you look at their genes, 60% of this small worm animal, those genes are overlapped with our human. That's why people study the gene functions on such a small model organism, which can reflect some of the functions those genes work in our own body. All right, that's genetics. Of course, we can modify their genetic structure. We can modify their genome. We can study each gene's function, etc. That's about genetics. And others, for instance, is also have a very short lifespan. It only survive 24 to 30 days from hatching from the egg until the death. So people use it for aging study. Right? You can study the whole development to aging process within a month. That's amazing, right? And also this small animal is very easy to raise. You don't need fancy incubators. You can just culture them on the table. I mean, its favorite temperature is around 20 to 21 degrees. That's the room temperature, right? And of course, if you increase temperature or decrease the temperature, you can kind of modulate the metabolism, so you can modulate the lifespan. That's why for aging, for development, there are so many studies. All right, just a small, just a brief introduction. The right-hand side shows some of those applications I have mentioned, but for each of the applications, it involves certain level of manipulation of such a small animal. For instance, for genetic screening, we have to modify the genome. Usually, the most traditional way, or the most popular way, is for using a microneedle or glass needle to inject transgenic structure material into the body. So that's a certain level of manipulation. And the neurodegenerative diseases, people always use high-resolution imaging to image the neural structures and how they change with time. All right? And similarly, all those applications involve certain level of manipulation. And you can guess most of the biologists that are doing it manually. So that's why manipulating such a small animal under a microscope, you have to see through the microscope. You need to use your hand to do manipulation. That's not easy. All right, so that's why with those motivations, we hope we can develop some robotic technologies which can facilitate or speed up or expedite the research in biology. All right, that's the overall motivation. So I will introduce those three topics one by one and give you a little bit more detailed motivation about each operation. So the first one is about injection. As I mentioned, in order to modify or change the genetic structure or produce transgenic worms, that means you are introducing some external genes into the genome, or you can remove some of the inherent genes from the body so that you can study the gene function to look at the phenotype. All right, so in that sense, you usually use the middle video shows you a manual injection process. You can see the diameter is about 50 microns. The tip of the needle is only 1 to 2 microns, and then you have to inject into a specific organ. You cannot just pierce into a specific a random location. You have to inject. You have to penetrate the gurnite where all those eggs are cultured and fertilized. So that your genetic structure will be able to modify the genetic structure of the next generation. That's why you have to identify such a small structure, and then you penetrate, and then you deliver. And that's why if you think about the whole process of manipulation, has so many problems. For instance, it's highly skill dependent. Any of you guys can do it. That's why, by the way, for those injection, any worm lab, based on our statistics, any worm lab, you need to do it. That's why if you study worm biology, probably one of the very first lab skill you have to practice is to do manual injection under a microscope. All right, it's not easy, but of course many students can do it. But the problem is the success rate is not high. I will show you some our data, compare the students and also some experts with the performance with our own robotic system. You'll see the difference. All right, so that's why our motivation is we really want to improve the success rate. We also want to improve the consistency of the operation so that we also the throughput. We can do, if you do an automated injection, of course, you can do a lot of injections, right? But if you do it manually, usually you cannot, like most of the operators cannot last for 1.5 hours or 2 hours. And you have to have a rest. But even after that, you come back, the performance will be not as good as the first time. All right, usually people, based on our collaborators' feedback, they usually don't do it over 2 hours per day. So the throughput has a limitation. I will show you some data later on. So those are the motivations. That's why the bottom line shows our summary is like how we do this robotic manipulation on a highly locomotive worm or animal, and then we can, so of course you can regard it as a robotic microsurgery, right? But of course now this operation is easy. It's just we need to deliver, we need to penetrate, using a needle to penetrate the worm body, specific organ of the worm body, and then we do delivery. So that still can be regarded as a surgery. So this is our first attempt. This is the first publication came out in 2016. It was done by my student, PhD student Peng Fei and Xian Ke. Those guys have become faculty, I think since 2018. So what Peng Fei did is, Peng Fei is a student working on microfluidics. So, but he's interested in worms. So I think, so those worms, you can see the left top video shows a locomotive worm. So in order to do injection, of course, some of my students have very crazy ideas, like let's track them. And then we don't do anything to immobilize the body, but just track them and we use the needle to follow and inject. But you can imagine it's not accurate at all, right? Because everything is dynamic. So that's why Peng Fei came up with this idea about how about we first immobilize the worms automatically. That means we can immobilize every worm sequentially using an automatically operated device. So this is a microfluidic device. On the right-hand side, you can see is a double-layer microfluidic device. If you're not very familiar with this type of devices, it's just a micrometer-sized channels fabricated on a rubber layer. And then you can seal the channel. That's why the blue channel are the fluid channel. We can load all the worms from this inlet, and this is a reservoir. We can store 50 or 60 worms. And then we have a drawing negative pressure from the outside, from the outlet, and then those worms will be sucked into a very narrow channel sequentially, because every time only a single worm can pass. But once you get the worm into the channel, this is a very it's not very long, but a very narrow channel. The channel width is just slightly smaller than the diameter of the worm. So once you get in there, this guy will be squeezed a little bit and immobilized. Of course, if you don't control the flow, it will easily pass through, right? That's why we have those red layer. This red layer is a pneumatic layer, not a fluid layer. It's a pneumatic layer on top. You can see on the bottom. This is the fluid layer on top. This is the pneumatic layer on the bottom. Once you pressurize this pneumatic layer, this thin membrane will bulge up and then to partially stop the fluid. That's why we can regulate the fluid flow using such a pneumatic microvalve. And we have multiple microvalves here. That's why once a worm is getting in here, we use computer vision to do real-time tracking to detect the presence of such a worm. And then we stop this valve, and then we stop this valve. And on the side, we have another valve which has to regulate the white channel, but this white channel doesn't have fluid, but it contains fluid. We have a needle inserted in there. Like before operation, this needle is outside the valve. This valve is closed because we don't want leaking from here, right? That's why we have to do a regulation. We have to do a vision-guided operation of the microfluidic device. So everything is operated. I will show you a video. All right. So once the worm is there, we close those valves, and then we open this valve. The needle will get in, and then do the injection. So only manual operation we allow the operator to do is to use a computer mouse cursor to click the location on the worm body where we do the injection. All right. That's the only thing. That's why you can imagine you're just sitting in front of a computer, looking at the operation of the device, and then you just click every time you have a worm present. All right, that's a process. I talk about a little in detail, but you can see the left-hand side is the whole flow, the whole device of coordination of the device and the robot. On the right-hand side is the video. You can see once it's loaded, the needle is getting in, it's injected, and load. You can see. This way is not very clear on the screen, but you can see a deformation. You see this deformation of valve. This is a closed state, this is open state. All right, so everything is regulated using a computer. All right, so that's how we do it. So we can sequentially inject 50 to 60 worms per batch, and then to perform injection. This is the first time. I was very excited because it's the first time we kind of operate a locomotive animal to do this type of delicate robotic manipulation. But the problem is still there, is I talk about this garnet, the specific organ we have to inject, right? But for this device, we cannot do it. That's why we try to use this device to do drug screening, because by injecting drugs, you really don't have to target a specific organ. You just need to target a specific area. That's why we try to do that, but it's not very successful in terms of that application, because worm-based drug screening injection is not a major dominating method. All right, that's why with this work we published a few papers. This is performance. You can see in terms of speed, we can inject 7 worms per minute, because you can see the whole loading is automated. So the only time-consuming thing is we click the button and then you get into injection, right? So the manual injection is you take 4 minutes to inject 1 worm. And then we also compare the success rate. So here the success rate we define is we observe the fluid delivered into the body. We use fluorescence, fluorophores, so we can see, observe them afterward, and then we count how many worms contains the fluorophore. So we could achieve almost 80%, but usually the manual manipulation is up to 30%. All right, so this is a comparison of the performance, but in terms of transgenic worm generation, it's still not the right system.
If you allow a question now, why is it 80%? Because this makes it seem like you should catch every worm like that, because they would go through the pipe eventually.
That's a very good question. The major failure the major potential failure is when we do the injection, not every time the material is delivered properly into the body. There are a few reasons. First, the worm body is squeezed a little bit. If you look at there, if you look at this one, you see there's something coming out. So sometimes those debris from the body will block the needle. That's why we don't have every time a successful injection, but most of the time. That's one thing. Actually, my student, I have a master student recently working on how to prevent this type of clogging of the needle using vision, using learning, and etc. But this is a practical factor. There are some other reasons, but this is a major one we found. All right.
How do you isolate one worm among many in the worm loading state?
You mean on device?
Yeah.
It's a long story. Long story short is we have to make sure all the worms has the same size, roughly the same size. So this is called synchronization. So once you use, anytime you use the worm to do a biological study, you want them to have the same age, right? So maybe 3 days after hatching. That's why every time you have to do synchronization. So people do synchronization using just visually under a stereo microscope. You just observe roughly the size of the worm, and you pick the ones with similar size. So after that, we load all those worms with similar sizes into this into the reservoir, in the loading reservoir, and then we just draw them because the structure of the fluid channel has a taper. So with that taper, only at one time, only one worm will go through. So you don't have to do a specific sorting. But if you are interested in sorting, I will show you another example. We do have sorting devices. We also have synchronization devices. We use vision to track the worm swimming in the microfluidic channel, and then we store them based on their size. I didn't include that work in here, but we do have some other, have that as well. All right, just go back to here because that's the first one. Again, just the carry-on method is we cannot do gurnard injection. This is the second generation. This is recent, very recent. My student took him, Peng Pan. He's now a full professor in China. Peng spent almost 4.5 years to finish this work. It's very time-consuming. So our idea is we need to do the gurnard identification. That means if I'm the worm, where the gurnard is this is the tail. The gurnard is on the both sides of the tail. It's a very long tubular structure going through both sides of the tail. We have a major gurnard. We have what we call it? I forgot the name of it. If one is longer, we call it major. The other one is maybe it's minor. But this may not the right terms. That's why you have to identify one of them you inject in there. So in order to do that, you can see the worm orientation. So the axial orientation makes a huge difference, right? So when we have to make sure the gurnard face the needle so that we can penetrate, right? So that's gave us a lot of challenges. So Peng spent a lot of time to try. Finally, we decided we have to give up the microfluidic solution, although it gives us higher throughput, because the loading of into the channel is random, the orientation is random. So what we do is, instead of using a microfluidic channel, this is still a microfluidic channel, but it's an open-ended channel. We use those side channels, as they are all connected to a single inlet. We use those side channels to suck a worm, to immobilize a worm. That's why once you have negative pressure, this is roughly the length of the worm, and the worm will be immobilized in there. And then we also found a mechanism we can rotate the worm along the axial, the longitudinal axis, so that we can do rotation. All right. So with that, we use this holding device to hold individual worms. We use a needle to inject them, and before the injection, we do the rotation through the coordination of this device and the substrate. I will show you the mechanism in the next page. All right. This is the new setup, and also we feel this is a proper setup because if you go to any worm lab, their injection system just look like this. They may not have all those motorized manipulators. We call them robot, but it's basically a computer controllable micromanipulator. They have some manual manipulators, and they don't have this device. They don't have this self-holding device. All other setups are the same. That's why you can imagine in terms of practical usage of this system in the biology lab, we only need to replace their manual manipulator with motorized ones, and then we gave them a consumable microfluidic device for holding the worm. All right. Of course, we have to give them the whole control system, but in terms of hardware cost, it kind of minimized. All right. That's the new setup. This is a sequence of the worm injection. So we load the worms in the middle. we load the worm here. This is the injection pipeline, and then we pick up a single worm using the device, and then we do the injection, and finally we put the injected worm in a separate area, so we can do the sorting automatically. All right. So in terms of rotation, I'll show you some videos. So this is how it works. So you can see we have a taper on the side wall, so that we can easily pick up. This is the cross section of the worm. It's along. This is the longitudinal axis. When we suck the medium in slightly, we will be able to immobilize it. But suck unexpectedly, Peng found out if you don't apply a negative pressure, by gravity, there is a very small fluid flow coming out of the needle, coming out of the device. And he found out this fluid flow will be able to rotate this worm along the longitudinal axis. And then Peng says, okay, I have some rotational mechanism. So he controlled the substrate. The substrate is the motorized stage. He controlled the stage so that with this fluid flow and with motion from left to right, you will generate a continuous torque on the worm body, and then you can control the injection, the orientation very precisely. Recently, Peng has a paper submitted to TRO. They can achieve around 2 degree resolution. That means the accuracy of the rotation will be around 2 degree. All right. So this is the rotational mechanism. Of course, we can by controlling by fixing this fluid flow, and then by controlling the substrate, we can rotate the worm. You can see this is the continuous rotation. You can see the substrate is moving step by step, and then we can rotate the worm. You can see there are some hollow areas. Those are some organs, and the gurnard is here. You can see there's some tubular structures in there. And once we rotate, we use the vision feedback. Once we tell using vision, okay, the gurnard is facing along this direction, and we just suck them onto the device, and then we do the injection. All right. So this rotation mechanism in our first publication in the NC, we actually didn't report all the details of the mechanism and modeling, etc. But we have a follow-up work we will publish very soon. All right. This is the rotational mechanism, and then we do injection. Okay, here is injection. So I'll show you this whole injection process. You can see the right-hand side is the video how we do the injection. We already immobilized the gurnard, and we switch the magnitude. Here is the gurnard, very small, very narrow. And the needle is getting in there and do the injection. And this whole process, still we leave the computer mouse clicking as a single only manual operation. So we allow the user to click on the screen. The reason is that we can definitely use computer vision to do that, right? And also recently we have those robust, like large models to do the learning-based recognition of the gurnard. But you can never, I should not say that. It's very hard to achieve high, like a 100% success rate, right? Anything failed, the system will run crazily. That's why we keep that as the only operation, manual operation. All right, so that's the injection. Once we have this injection system operating properly, we compare our robotic system performance with manual orientation, manual injection. So we actually try 2 types of injection. One is a single gurnard. That means we inject only one side of the gurnard, one side of injection. We also have the double gurnard. That means we inject once, we rotate by 180 degrees, we inject twice. And we compare their success rate. You can see double gurnard gave us a little bit higher success rate. And also, because in order to create transgenic worms, we need to produce enough number of offsprings. Usually transgenic worms, we have to culture the next generation and the generation after that, so the second generation. Because once you have a stable expression of this genetic feature, in the second generation, we see this is stable. That's why we count the number of progeny, the number of offsprings every time. You can see double gurnard give us almost double the number of next generation worms. So that's really nice. That's why for some of the biology studies, we use double gurnard injection. But with this double gurnard injection, you can imagine manual manipulation will be even harder, right? You have to do rotation, sucking rotation, and then that's why robotic is more desired. All right, so skip some of the technical details. I'll show you some results. So once we have this system, by the way, our biological collaborator is Professor John Krakow. who is a professor in system biology in our UFT. So John is the best manual injector I have ever seen. So he has 22 years of experience, and John actually screwed up our results a lot. Because I expect our performance will be much better than the human. But John is this guy, oh, where is this? Yeah, you can see we compare 3 experts, those are 3 experts, 15 years experience, 22 years experience, this is John. And then 7 years experience. Sorry, maybe I should not disclose his identity, but anyway, this is internal. You can see our robot, in terms of the number of offsprings, our robot generates the similar number of offsprings with the 3 experts. John is the best, so he insists us to include his own manual data in there. He said, okay, but I told him you're not really your performance is not really a common performance like other people. But he said, okay, I'm the best, I will just get in there. So that's why our conclusion is our robotic system in terms of quality of transgenics is similar to the best experts. But if we compare with students, I asked John to give me 6 students, and those students, some of them in their years, 1 year, 2 year, and half year experience. And with all those students, our robotic system gave a much better performance in terms of success rate, in terms of transgenic quality. All right. So interestingly, we also show this robotic system gave any new user a very short learning curve. That means if any of you guys want to use the system, I can introduce you to my student. You only need to do, this is the number of trials using the system. So those are 5 new operators. By operating the system by 1, 2, 3, 4, you can see the performance finally is all increased to around 80 to 90%. That's a very decent, a very good performance. And then at the 4 times of trials, we look at the number of offsprings. You can see all the 5 operators with the control operator. This is our very experienced folks. And those are 5 people using our robotic system. This is the manual performance. You can see all of them has very consistent performance. This shows you the robotic system is very easy to operate. You only need to try a few times of operation, and then you can achieve a very good performance. This is really some of the amazing results I personally feel this is coming out of the robotic system. It's really not beat the best people. By saying that, because I cannot beat it, I cannot beat John, but it gave you a very short learning curve. You don't really have to do a few months of training, and you can do a very decent job. You can do a much better job than a new operator. All right, those are the results. In terms of biology, I will not talk about too much details, but we work with John to find a biological study where John is studying the alternative slicing of the gene expression. What alternative slicing means is a single gene could generate multiple proteins, and the whole biological process of leading from one gene to multiple proteins is called alternate splicing. So John is studying some of the specific proteins, what is the governing mechanism of the alternate splicing of those genes. So in order to do that, like long story short is you have to do multiple combinations of different genes and then do the transgenic study to look at their phenotypes. So we're talking about, you can see, we're talking about 200 genetic combinations. We performed over 1,000 worms. This is a medium scale. This took about, including the follow-up studies, took about 2 months. But we had a 5-year CHR grant with John. We are trying to target to do 20,000 combinations. So that's why John, our system haven't delivered yet, but our target is we wrap up our system, we deliver a whole system to John's lab to allow their students to do it using the system. All right. So this just shows you we can produce a very reliable biological results, and also we have the first time found a new evidence showing this specific RNA binding protein regulates the alternate slice of this Zoom1 mRNA. Of course, this is a very small data. It's not really reflects a lot, but you can imagine for such a large scale biological study, the robotic system will make a huge difference. All right, that's just a proof of concept operation. All right, this is the injection. I think it took a lot of time on that because I think I really like this story because it really shows you the specific challenging about working with those small worms, and also what is the major difference our system makes, right, in terms of the performance. All right, so that's why we have this 5-year CHR grant. Actually, we recently collaborated with some from US folks. They're applying for NIH grants on this, using this system. So we're also hoping to make the whole system robust as possible, and also to eliminate the human operation as much as possible. Those are the major next steps we want to do in terms of technology. All right? Okay, so let's go to the next one. Is about automated characterization. This is actually a side project produced by Peng, but we have been working on this operation for a long time. We want to do 3D high-resolution imaging of the fluorescent signals in the worm body. So if you use a confocal microscope, you know, for thick material, you have to do a scanning, but very often once the light penetrates from the bottom, penetrates a thick material on the top of the material, the fluorescent signal will be lower. Right? That's why even though you can do 3D reconstruction using confocal, but sometimes this is limited. But if you rotate by 180, you can image the bottom half first, and then you rotate the top one facing down, and then you image another half. All right. So this is the major motivation: is we want to do high resolution imaging, and that's why we have to do rotation. Of course, if you think about the way we talk about this story, we're really doing it in a reverse way. First, the Peng can do the operation, the rotation, and then we think about, okay, what we can do with it. We have the hammer, but I talk about this in the reverse way. That's why you can see this is still the injection system. Peng is still using the operation, is still using the rotational mechanism. But Peng has a detailed dynamic modeling so that we understand the whole dynamics of the system. And then we also do some controls to precisely control the rotation. And that's a new work we just submitted. And then that will allow us to have a precise operation, because if you want to do 3D reconstruction, the current orientation degree angle, you have to give very accurate results, right? So that's why we do this operation. They also show, you can see we can rotate thinner, smaller worms, medium-sized worms, larger worms. We can also rotate egg of the worm. So this is around 1 millimeter. This is about like 10 micron. So larger scale, smaller size object can do it. So we can do the rotation because it's all like a longitudinal shape, right? So that's why we can do that. Once we have that, what we first do is we do rotation plus 3D reconstruction. We can look at the neural structures, especially the detailed dendrite here. So those are the bifurcation here. Those are some of the details neuroscientists want to image. But using confocal, most of the time is working, but we can actually show a better imaging quality. So this tool, we just do a hand waving like a proposal is it could be used to do disease studies about degenerative disease. But we didn't do much of that along that line. And also, besides the molecule structures, we can also look at the morphology, the surface morphology of the worms, and then we do 3D reconstruction. This whole 3D reconstruction is common techniques. We didn't do anything new here, but with using this whole framework, we can reconstruct a 3D dimensional structure of an embryo or a worm, and then one step we need orientation is here, is the registration step. It's like we have to do the worm alignment, we have to record all the angles of each frame, and then we can do a 3D reconstruction. That's where we use the rotational control, all right? So with that, this is a dynamic video, shows you how we do the 3D reconstruction. You can see, because we have high resolution of the imaging, you can see some of the textures here. We can observe This is around 5 to 6 microns, this is around 10 to 15 microns. We can observe very detailed structure on the surface. I talked to many biologists. They said, okay, this is cool, but they don't have any clue about the surface texture, what that is correlated to, because they never be able to do it. Or maybe, I should not say that, because they can do confocal, but they never pay particular attention to those features. We're hoping we can do we can use it for some biological implications. All right. So actually, in order to publish this paper, Peng did a little bit biology study. I would say this is very shallow, not a very deep biology. But Peng actually look at, it's still under the context of the alternative slicing. He studied a few mutants with those genes like knocked down, so that you can observe their morphological difference. You can see some of them are longer, some of them are shorter, some of them are wider, some of them are thinner. And then by looking at the statistics of all those features, and we use those as a morphological phenotype parameter to study the genetic interaction. But again, this is not the determinative measures, right? But we do give the biologists some additional features they can look at or they can consider, right? So this is, but this work is still a more technology-focused study. But we are hoping maybe by publicize or make those codes public online, because the worm biology community are very open to new technologies. Hopefully they could use our work codes to do some of the studies, but that's something we are still exploring. All right, so, okay, on the third part, the second part of the characterization is more on microfluidics, because some of the folks ask sorting. This is a sorting device. You may ask, you may think, okay, I'm basically throwing to you a bunch of technologies related to worm biology, to worm manipulation, but this does have a requirement. This requirement or this need, biology need, actually I got this information since, in 2010, when I was a postdoc in Harvard. Actually, some of the biologists are studying development of biology using worms. They want to do imaging, like a long-term imaging of a single worm egg from laying, after being laid until to adult worm. So that is around 4 to 5 days. But the first thing is very hard to catch such a small egg. Usually how people do get those eggs, they have those adult worms and then they lay eggs, so you will have a bunch of mixture of worms, larger worms, smaller worms, eggs, and some debris, and then you pick up using a very small needle or some small like a filament to use adhesion to pick up a single worm. Very, very difficult. So that's why we thought, okay, let's do some sorting, because sorting in microfluidic is very easy. This is also what Peng did, is it's a very simple device. In the inlet is the inlet here. This, in the center is the inlet. This is a spiral channel. If you look at cross section, this is just a rectangle channel. But once you have this spiral shape, you flow out of the fluid. You can see, if you look at the fluid flow, it's called a Dean's flow. You have this circulation at the cross section. This circulation will help us to sort the small particles or the eggs to this side and leave the larger ones on this side. Of course, based on the size, and you have the largest object here, you have smaller worms here, but most of the eggs will reach the other side with going out, all right? So this is the channel. On the sidewall of the channels, because the eggs are close to here, we have a side channel which can have a small flow going this way, going to the next channel. So though all the small eggs will reach, each single egg will reach such a side channel, so that finally we will have around 40 to 50 eggs arranged on this spiral channel. So you can see this is the whole image showing you where this is the worm, this is the small egg. Finally, this egg will get in there and get captured. All right. Once we get captured, we will do a long-term imaging. We can do automatic scanning. Periodically, we image them. I'll show you a video. You will see how such a worm. This is a sequence. I'll show you this video. You can see this is the time how a single cell will develop. This is embryo, it's not a cell, it's a fertilized egg. How it will develop from an egg, from an embryo into a small worm, and how it hatch. You can see it's almost in the shape of a worm, but still encapsulated by the membrane. And finally, it will reach, it will hatch, and it will get out, and then reach the small young larva, a small larva stage. So that's this time. You can see we can do, by capturing those individual eggs, we can do the long-term imaging to observe their morphology. We can also do fluorescent imaging to look at their fluorescent expressions, right? So that's a sorting. Finally, we can reach the whole process. So 8 hours, it will take around 11 hours. It's almost there. You can see it's moving, finally it will hatch. All right, so, still not there yet. Let me drag it. Sorry, I cannot have the bar. But anyway, you just see, you can look at here, this is the hatched one. That's a hatch. Hold on. Oh, sorry, I missed that part. But this is right after the hatching. So anyway, with this, this is the one demonstration. We say like, okay, what we can do is this. So we did a very simple study. Is we change the salinity level. That means we change the salt concentration in the culture buffer. We found out once you have a high osmotic solution, and then it will make the development temporarily arrest. And then once we replace the high concentration culture medium with a regular culture medium, it will resume. This is just only one thing we did, because microfluidic channel, you can flash different solutions once you have doing the culture, right? And you can imagine during this development, you can treat the egg with different solutions, and you can do this type of large scale or medium to large scale biological study. This is another one. So this is more microfluidics, but we do have automated scanning of the stage, but we don't have much of the manipulation part, but still a fluidic manipulation of the worm. All right. So that's the second part about characterization. I'll reach the last one, because the first two parts, we mainly focus on some specific structures inside the worm body, right? Or sometimes it's the whole worm morphology. We look at the morphological parameters. But because we work with the worm for a long time, I was always imagining, because the worm, you have seen the video, is a kind of snake-like locomotion, right? So there are two locomotion types of the worm. One is you culture the worm on an agar plate. Is a white, solid, not very solid, soft agar surface. It will locomote on it. This is where you see the video. The second locomotion pattern is it can swim. But swimming will be different. Locomotion is a snake-like locomotion. Swimming will like a single motion. It's like a bend this way and bend the other way. Bend the other way. So a single curvature, positive, negative, and then it will swim in the video. So we focus on the surface locomotion behavior because it looks like a snake. And then my idea is very intuitive. It's like, okay, this looks like a snake, but in robotics, we have so many bio-inspired design or biomimetic designs to mimic the locomotion of the snake, right? And the major challenge is how we design or integrate all the actuators, sensors, and etc. to achieve a very snake-like locomotion, like jumping, and etc. Right? So that's the major challenge. So my idea is, okay, this is a small worm, especially at the micro scale. What if I just control this guy? So that means the activator, the structure, the robot structure, the activator and sensor are all in the same living animal. So my robotic component is just I control it. So it's a very intuitive idea. Of course, even you can fully control the locomotion of the worm, it's still not a robot completely, right? So you have to show some capability of manipulating. But the first attempt started at 2011. So this is a really long time ago. But we published this paper on 2020. So my student Xianke, who also developed the robotics, the first version of the robotic injection system, he spent almost 5.5 years, including some of his postdoc time, to achieve this like a first attempt. So that's the natural locomotion. Our idea is very simple. This is the structure of the worm. This is the head. This is the tail. So all the neural system, the whole head contains all the neural system, the whole neural system. So the neuron is connecting with the side muscle cells. The blue areas are the muscle cells. There are so many muscle cells connected together. And the neural system gave an instruction, gave a signal to the muscle cell. The muscle cell will bend sequentially from the head to the tail. You can imagine it's a wave propagation from head to tail. And that wave propagation determines how the worm locomotes, all right, on the surface. All right, that's the mechanism. So what we did is we first enable the neuron to be controllable using light. So this is simple. Actually, it's called optogenetics. It's basically we introduce a light-sensitive ion channel on the surface of the neural muscle cells. And then once you shine a blue light on it, the ion channel will open up. It will pump calcium in the muscle cells. And then that higher concentration of the calcium will make the muscle contract. So that means if I give a light, it will contract. So this is the first, we use optogenetics to make the muscle cells controllable. And the other side is, I really don't want this neural system to control the muscle cells, right? So we actually use a chemical to temporarily disconnect the connection between the signal connection between the neural cells and the muscle cells, so that the whole worm is not moving anymore because the muscle cells are not controlled by the head. So we are basically paralyzed the worm. All right, those two measures, that's what we do. So once we have that, you can see usually the worm will, now it's locomotive, but the paralyzed worm will be a street worm. So we show that we shine light to a part of it, and that local muscle will bend. The bending curvature is proportional to the light intensity. Look at this data. So the bending curvature is proportional to light intensity. That means we can use it as an activator, right? But the challenging part, it took us almost 2 years, is if you study like a snake robot, probably you know the mechanism, the locomotive mechanism in the snake is you have a sinusoidal wave body curvature because it's snake-like locomotion, and then the muscle contraction also form a roughly sinusoidal wave. Some of the muscle, if you look at here, this is the muscle contraction. This is the curvature of the worm body, and those intensity, those fluorescent intensity shows you the calcium level in the muscle cells. You can see the highest intensity shows you the largest curvature. So you can see those largest curvature also shows roughly a sinusoidal wave, right? But this sinusoidal wave has a slight phase difference with the body curvature. This is the mechanism for snake locomotion. It's also the mechanism for worm locomotion. So we first observe using this study, we do the fluorescence imaging of the calcium, and we also check the snake references. But it took Xianke a very long time to build up a simple kinetic model and also a dynamic model to understand how this works. So finally, our conclusion is those two curvature sinusoidal waves has to have a phase difference. So with that, we kind of understand how we do it, because we just need to give 3 light patterns on the curvature. Each of the light pattern will make the curvature, the muscle cell to bend locally with the maximum curvature. And then we start from the head, and then those 3 sinusoidal waves will start from the head, and then start to scan from the head to the tail. But we try to maintain, we use vision to track this body curvature. We always maintain a fixed phase angle difference between the activation light sinusoidal wave and the body curvature sinusoidal wave. All right, that's how we do it. I'll show you some videos you can see. And also, in order to, this is the street locomotion. In order to try to make the worm to turn left or turn right or suddenly turn with our omega shape, and then we have to regulate the intensity, the activation intensity. You can see the activation intensity has to be different. With that, we can achieve, this is the natural worm locomotion. Those are not paralyzed worms, locomote natively. Those are the paralyzed worms controlled by the robotic system. And then we can achieve all the 5 types of locomotions to mimic the natural worm. All right, so one thing I want to show you is, actually that's a follow-up work we did, is you can see with the straight line, the intensity on the left and the right has a huge difference. Why is that? Because when we do the transgenics, the ion channel distribution on one side of the body is quite different from on the other side of the body. That's why our first transgenic worm, the activation property on 2 sides are different. Actually, it took me another master student for 2 years, Zhao Yi. She's now in Michigan. Actually, Zhao Yi developed a more reliable mutant, which gave you uniform light intensity control. That gave the control system easier task. But anyway, this is our first report. Those are the videos you can show. So basically, you can see this is the head. So those light intensities scanning from the head to the tail. Once this guy is gone, and another light will come up, we'll continue scan. Well, of course, we regulate the intensity on both sides. We can control the locomotion. Of course, during this whole control, we have the vision feedback. So that's the control system. We have the vision feedback. We know where the worm is, what is the curvature, what is the sinusoidal curvature, and also we can do closed loop control of the direction and location. So here is the position control. You can see from all 3 worms, from A position to B position, they start with different directions, but finally we can all control them to B using closed loop control. The controller is nothing fancy. This is just regular PID controller. But with the feedback, the closed loop feedback, we can do that. And also we can control the direction. You can see all 4 worms, starting with different directions, we can all control them to horizontal directions. All right? So actually we can control, to summarize, we can control the position, while we can also control the direction. That's all we can do. All right? So finally, because we published on Science Robotics, the editor likes this work, but one of the reviewers give us very harsh feedback. So this is not a robot because you are basically just control this. But our title is also locomotive control, optogenetic locomotive control of C. elegans. We never talk about the worm robot, but of course in the introduction we talk about this as a robot worm. Hopefully eventually we can make it. That's why this is the only demonstration we have. So let me replay the video. Actually. So we have a maze here. We allow the worm start from here. We do the path planning in hand. So we control them from one position to another, and then eventually the guy just to navigate the worm. But you can see we do miss a point. The last one, you can see it reach here perfectly. This is perfectly, and then this is perfectly, and then we miss the next one. You see, there's one sudden change. We miss this one because at that moment the worm itself is not completely paralyzed. It naturally turns the head a little bit. So that's a disturbance of a controller, right? But the closed loop controller, the amazing part is after you miss that point, and then you can reach the next point because it will correct the disturbance, right? So this is the only demonstration we have. Okay, we kind of control this to do a maze navigation. That's it. And then the reviewer asked us, can you do some manipulation? We did try. We tried all kinds of things. We attached some micro objects to the head of the worm so we can make it to deliver, or we use the worm head to push. I think the pushing strategy is promising because that's a typical robotic manipulation strategy, right? But of course, the head is not doing straight motion; it's doing this. So it's not easy for us to achieve this in short period of time. That's why finally we frankly tell the reviewer and the editor we cannot achieve manipulation at the moment. But we did some discussions. We think about maybe we could have some temporary attachment of the macro object to the head, so we can deliver the object from one location, one area to another, or etc. We even imagine we can make this worm to wear some ultra soft electronics, so we can stimulate the worm body to do some control as well. There's so many great ideas, but this is the first attempt. So published paper, I think I like this idea. Is the value is it provides people a relatively new strategy to achieve micro-scale bio-inspired micro-robot. All right? Of course, if you think of DARPA's work, before us, they have been using like insect to do robots, right? That's why it's along the same line, but at micro scale, soft-bodied worm, that's our first time we do this. All right, so I think it's almost, time is up. I'll stop now, and just as a brief summary, I think I show you some manipulation, robotic manipulation systems and technologies we have done on those micro-scaled animal. Personally, I do hope some of the technologies will go downstream to reach some practical biological applications. I think worm injection is definitely one. I put a lot of hope. Hopefully, my whole dream will be after 5 years, 3 to 5 years, there are many biological labs who are using our injection system. That's my dream. I really think that will make a difference for their own studies. We're also collaborating with some people to customize this system. But if like 50 labs are using my system, some of them are producing like Science or Nature results using our system, I think that will be our contribution to the field. All right, that's my whole dream. But of course, some of the robot worm work, I think is more like explorative. If you're interested, I can talk more about what could be some real applications that this system can be done. But anyway, with the time limitation, I will just stop now. And thank you for your attention. I'll be happy to take any questions. Thank you.
Take the other microphone as well.
You can do. You just have to hold it first.
Okay, this is for you. Okay. So guys, there are a bunch of questions. Let's start with you.
Xinyu, thanks for the great talk. I have 2 questions. So the first one is about the automated microinjection. Does your pipette have control over how much fluid or how much material it's injecting?
Yeah, we can control. We can easily control to a few nanoliter. You can control the volume down to tens of picoliters, because this is not what we achieved. Is you use a pressure regulator to give a very short path of a constant pressure. You can control the pressure level. You can control the width of the pressure, the time period of the pressure, so that you can regulate the volume. So you can buy such a machine. Actually, there are commercial products on that.
Got it. Second question, sorry if this is naive, but you mentioned CRISPR.
Yeah.
And it seems like microinjection seems like a very crude, not targeted type of editing, but CRISPR is very targeted. So how could you do CRISPR with this type of system?
I think to clarify, microinjection is the method to deliver the CRISPR material into the garnet. But this CRISPR material was designed by John Clarko, who is an expert on that. So actually the CRISPR material has to be very specific to target a specific section of the genome, right? So the material is synthesized. But those ones, when we do this system, it's still not very popular. John is one of the first people doing genome editing using CRISPR system on worms. But nowadays, I assume it's very common. Yeah, you can buy them through in company, or you can just get some from John's lab.
Got it, thank you.
Very exciting, touching very closely the future, the wishful think at least. I was wondering if you are aware of the research by Professor Ryu, RYU.
RYU?
RYU. I think first name is William. He's in physics and he's in Donnelly. And he's doing exactly with C. elegans, this curvature and a lot of fun.
Oh,
so you better be familiar with this fellow. He's doing it for many years now. My question or wonder is, can you deal with, I think you dealt with chemotaxis, but can you think about blocking metastasis in cancer?
When you say chemotaxis, you mean when we do the paralyzation?
Chemotaxis are the experiments that
Professor
Ryu is doing, all these gymnastics with the worm and fun. But the burning thing in my book is metastasis.
Yeah.
Can you imagine blocking the cell mobility, motility, and so on with a robot who is clever and faster and can tweak those cells? Because this is life and
That's a very interesting idea. What type of cells you're working on?
Pardon me?
What type of cells you're working on?
Well,
Neutrophils or some locomotive cells?
no. Well, it is locomotive cell. If you look at under the electron microscope,
Yeah.
you look at these movies of metastases.
Okay.
See the
Oh, yeah. Oh, I know what you mean. I know
cells,
what you mean.
and the cells are clever to go
Right.
through barriers
Right.
generated by
Mm.
two other cells that are slower.
Mm.
And these cells are big, and you wonder how they manage through
Mm.
the barriers. But there is lots of room to
Mm.
do work, to intervene
Mm.
and save people.
I see. I think optogenetics with the control of the light, using optogenetics may be one way, because you can regulate the local activity using optogenetics. I know people using optogenetics to study neuron cells. You can regulate, that's one way, but maybe you can talk more. I haven't think about in details, but a very interesting suggestion.
So it's very exciting to see your work, and I have somewhere between my
Sure.
ears a dream
Let's talk
that
after
we
this.
stop this metastasis because
Mm.
all these hospitals are
Yeah.
full of people
On neurons,
who are
on
at
cancer
the
cells.
edge, and
Mm.
so far medicine didn't find a way to stop metastasis because at that point it's all
Yeah.
game over.
Yeah.
But you, Professor,
Mm.
you may come up with Professor RYU
Mm.
and quench.
RYU, right? Real.
You are LYU.
William
and
R-
he is
RYU.
William.
Mm.
Yeah,
Okay.
so you know him.
Okay. I will search his name. Thank you.
Welcome.
Yeah.
Okay, more questions? Okay.
I thought the concept of optogenetics was really cool. You're using visible light, red and blue light, to open these calcium ion
Yeah.
channels, right?
Yeah.
So I can see how that would be limited in organisms that are bigger because
That's right.
of
That's right.
penetration depth of visible light.
Yeah.
Do you know if there are other methods similar to optogenetics that use maybe like near infrared light or even ultrasound to penetrate?
Electrical stimulation would be more, I think it could have a wider scope of application. You're totally right, because using light, optogenetics is more appropriate to thin layer of materials like adhered cells or maybe worms. But if you increase the scale, the penetration will be a major problem. But that's why for a larger animal, when people try to control the muscle contractions, they use electrical stimulation very often. That could be an alternative approach. That's why actually talking about applications, once we publish this Science Robotics paper, there's so many people calling me. I still remember a journalist from UK, from London, he told me, can you do this type of typical drug delivery applications using this worm? I said no, because drug delivery will get them into the body, right? First of all, our light will not be able to reach. And secondly, this worm, we cannot swallow it. Because this worm, the close relative of this worm is the parasitic worms. So that's why there's so many concerns, you put them into the body. But I do have a few ideas. One is, because the professor mentioned chemotaxis. Worm has been used for chemotaxis a lot, because the worm has a specific flavor of a specific chemical. For instance, like glucose. Usually it could chase after the high concentration of glucose. So that's a typical method for people doing chemotaxis studies. Basically, how they locomote towards a concentration gradient or away from concentration gradient. But this is their native flavor. You'll never be able to force a worm to move from their favorite concentration or chemical to a non-favorite location, right? Or temperature. The thermotaxis is another one, like a certain temperature. But our device or our system can do that. We force the worm, we paralyze the worm, so the brain doesn't work anymore. We can force the worm from a liked location or concentration to a disliked one. We can study the neuron, the response of neural circuit. Actually, that's something we are discussing with some neuroscientists using worms, but that's one biology application of this system. Another application is we want to use this small-sized snake-like locomotion model to do micro-scale modeling of the worm locomotion, like how its body interact with the substrate. That's another, if you understand this at the microscope, hopefully, maybe not eventually we can achieve that. If we can study, we can find out some different mechanism of that microscope. Maybe it could help us to design some microscope robots. So that's a more fundamental study along the robotic line. All right, those are the two applications I can think about. That's why once we publish this paper, there's so many people asking us, because micro robotics, people you do drug delivery, do like microsurgery in the body. We say no, our technology cannot do that. But maybe those are two directions we could use it for something, right?
Okay, any more questions? Yeah, I'm coming.
Yeah, I'm just wondering, how do you guys actually implant the receptors of the Optogenetic?
It's actually injection.
Oh.
So it's transgenic worm. So we create this transgenic worm using a complicated process. It's not like a single time injection. You have to culture the worms through multiple generations. This whole process took us around typically 3 months. And starting from the design of the genetic material. We are not doing this by ourselves. We are collaborating with Professor Zhen Mei in Mount Sinai Hospital. She is one of the top experts on transgenic studies on worms in Canada. That's why once, because Professor Igor said I moved from McGill to here. Before, I didn't find any proper collaborative labs on this. Once I moved here, actually, May's lab helped me to achieve this. But again, this generation of this transgenic worm is not trivial. It takes a lot of time. Yeah.
Okay, I think we're at time, and we will move the conversation to the coffee.
Sure.
Thanks so much for this wonderful talk. It was really exciting.
Thank you so much. Feel free, I will stay for a while so we can talk. All right, thanks.